Respected judges, Assalamu alaikum. Today we represent Team TMV Tech Titans competing at the high school level for Teknofest 2026 Technologies for Humanity competition. We are proud to present our solution for healthcare professionals, the Biosense script. My name is Mohammad Rasul Dishan and standing alongside me is my teammate Muhammad Moin Farouk. We will be your host for today. Let us examine the scope of our project. Centralized lab tests like PCR fail during remote outbreaks because they're expensive, require steady electricity, and need refrigeration. This causes fatal diagnostic delays. We solve this by extracting non living cell machinery and freeze drying it onto standard cellulose paper, regulated by synthetic logic DNA gates. As shown in the diagram, it detects specific viral RNA instantly via a clear visual color change. This brings laboratory grade accuracy directly to rural clinics, matching the health and well-being technologies theme.
Our primary purpose is stopping the dangerous diagnostic delay. Right now, remote communities wait days for biological samples to travel to urban labs, letting local outbreaks grow in regional epidemics. Biosense Strip works with the physical simplicity of a pH paper but keep the high accuracy of centralized PCR test. Giving immediate data on-site allows for exact quarantines, which prevents mass livestock losses in farming and cuts down the massive capital drain of national pandemic responses.
The root of the problem is that accurate testing tools are too fragile for field use. Current high fidelity protocols demand an unbroken cold chain and steady grid power, which offset grid zones simply do not have. This creates a stability cost paradox. Cheap antigen tests are inaccurate, while PCR tests are fragile and expensive. The medical world currently lacks a hardened diagnostic node, a system tough enough to travel in a backpack through 40-degree heat without spoiling, but smart enough to read specific genetic sequences. The biosense acts as this rugged node to fix this diagnostic gap.
We solve this paradox using synthetic biology and cell-free computation. Our molecular architecture rests on three pillars. First, we build custom DNA logic gates called toe-hold switches. They sit in an off state by default and unfold via branch migration only when they are bind with a perfect target RNA match. Second, we create a cell-free computing nodes. We extract just the ribosomes and translation machinery from the cells, making a stable liquid circuit outside an organism. Third, this cell-free design ensures zero mutation risk because there are no living cells. The biological sensor cannot grow, change, or dial during extended field storage, making it completely stable in the field.
Biosense functions exactly like programmable software. If a virus mutates, our physical manufacturing machinery remains untouched. We simply recode the target sequence into silico and print updates onto paper strip in days. This allows massive global scalability. An entire modular molecular lab is condensed onto 1 centimeter strip of cellulose paper, bringing manufacturing costs under 50 cents. Best of all, it features built in color logic. Target detection triggers the synthesis of a reporter protein that shifts from yellow to deep purple in minutes, giving an easy yes or no answer without electronic readers.
We brought this concept to life through a two-phase engineering pipeline. In phase one, circuit designing in bioinformatics, we used computer modeling to map local pathogen strains and design a toehold triggers, optimizing binding strength to eliminate false negatives. In phase two, cell-free optimization and substrate integration, we moved to the wet lab. We extracted cellular machinery from bacteria, perfected freeze drying protocols to bond it to paper, and built a protective user interface to keep reagents stable against ambient humidity before use.
Our phase 3 bench testing confirms our molecular logic works flawlessly. When exposed to the target viral RNA, our sensors hit a remarkable 96.8% detection accuracy. When tested against mock stains or plain water, the switch stayed completely closed, proving zero cross reactivity. The visual color change triggers accurately at an ultra low concentration of 45 femtomolar femtomolars within exactly 18.5 minutes. Furthermore, degradation tracking proved zero loss of enzyme stability at 40 degrees Celsius for over 180 days. Keeping our operational margin of error under 3.2%, we have built a test that survives backpack transport in blazing heat.
By switching our diagnostic architecture from silicon chips to paper biology, we completely bypass fragile microchip supply chains. Production relies entirely on high volume, standard paper processing techniques, creating a 95% reduction in unit cost. While a standard lab PCR test costs around $15, a single biosense strip costs just 48 cents to make. This sub 50-cent price point makes high fidelity screening for affordable for government emergency programs, international NGOs, and low margin flaming sectors globally. It turns expensive testing into a cheap commodity.
True innovation must be environmentally sustainable. Biosense strip features a biocompatible life cycle built entirely on biodegradable paper, leaving a negligible footprint compared to liquid chemical waste. This platform eradicates plastic waste by replacing millions of single use plastic PCR tubes and lateral flow casings. Additionally, our freeze drying technique achieves a zero infrastructure impact by removing the cold chain, slashing carbon emissions from refrigerated shipping networks.
Our platform serves four distinct target sectors where current diagnostic tools fails. First, rural healthcare workers gain instant triage capabilities in off-grid outposts. Second, livestock farmers get early warning tracking to catch agriculture pathogens before they force mass herd curing. Third, disaster relief teams can deploy this drug screening platform to isolate infections in crowded camps. Finally, national health agencies gain a low cost way to stockpile millions of tests.
The ultimate value of our project is public health security. By moving diagnostic tools directly to the point of exposure, we compress the response window and turn regional health strategies from reactive damage control into a proactive containment. This allows for hyper localized quarantine protocols instead of forcing entire cities into costly lockdowns. Economically, it prevents heavy losses in farming, and environmentally, it provides zero power testing that degrades naturally back to the earth.
Our development timeline was fast, structured, and entirely completed across 2026. We started with bioinformatics, modeling, and trigger design from mid June to mid July. Next, we spent late July optimizing the cell free extracts through most of August. We worked on freeze drying the solution onto paper and running humidity stress tests. We finished by executing full sensitivity trials against viral mimics.
Evaluating our project honestly through a SWOT analysis, our strengths are the low 48 cent unit cost, zero cold chain leads, and zero false positives from our genetic match requirement. Our weaknesses, on the other hand, include faint visual lines at target levels and production complexity regarding drying climates. Opportunities lie in recoding modular DNA software for new variants in days, and our main threats are breaking all medical assumptions that cheap tests are fragile and managing biohazard disposal protocols in remote areas.
In conclusion, BioStrength Strip replaces a fragile electronic hardware with smart cell-free paper biology, putting laboratory accuracy right into the hands of frontline workers, reclaiming public health by democratizing the logic of life. This is Team Tech Titans signing off. Thank you.